for not only giving me the opportunity to speak, but also being kind enough to move around my time slot. It's 1 a.m. here at the moment, and so I'm not only hoping to stay coherent, but also praying very much that I don't wake up the kids. So we'll see how that goes. And so in the last session, so I should preface this by saying that I'm not going to be talking about stem cells at all today. But hopefully what I talk about is somewhat applicable. And so in the last session, we heard a lot about highly multiplexed imaging technologies, in particular, imaging mass cytometry. And so our university purchased a Hyperion imaging system a couple of years ago. And so as a statistician, I've been getting slowly inundated with more and more IMC data. And so because of this, while there's lots of challenges around analyzing these highly multiplexed images, today I'm going to be speaking about some work from a really talented med student, Nick Kenyette, about how we've been going around analyzing some of the spatial components of this image data. And so specifically, we've been looking to see if we can develop methods to ask a really basic question, and that is the localization of different pairs of cell types changing between diseases, whether that's controls versus disease, or response to treatment versus not response to treatment, no response. And so while you can obviously ask questions about whether cell proportions are changing, the real big power of these technologies is really trying to understand the spatial orientation of the cells. And so when we started doing this, we realized that there's some key challenges. So for starters, for technologies like IMC and some of the other multiplex technologies, the images really aren't that big. And so for IMC, you're talking about 1 millimeter squared, maybe 2,000 cells. And if you're measuring lots and lots of markers, which is the great thing about these technologies, you also have the capacity to start really deeply phenotyping cells and potentially splitting them up into more and more cell types. So you can imagine that if you've been able to identify 50 different types of cells, if you've only measured 2,000 cells, that cell number starts to drop quite quickly. And so that creates some problems in how you're analyzing things. Also, because these images aren't quite big, to get a really good picture of a person's tissue, people often take multiple images per person to really get an understanding about the heterogeneity within a person. Another challenge that we kind of faced was what distances to look over. Obviously, sometimes you want to look at really close cell interactions, and sometimes you might be interested in things that are slightly more distant, cells traveling to certain areas. And then also tissue tends to have structure. And so this structure can change from image to image and person to person, and sometimes this can confound the kind of conclusions that you're wanting to make. And so this was something else that we saw as a challenge. And so to get around this, we decided to analyze our data as a point process, that is taking our cells and reducing them to a single point in space. Obviously, we throw away data here, but we think it's worth the sacrifice. And so we've analyzed these things as a point process model, and with that we can summarize the localization information with a K function. And this is just a fancy way of really taking the average number of cell type B near cell type A, and then scaling this so that that localization is comparable across images of different sizes. We can scale this further so that we end up with something, we end up with a variance stabilized version of this measure, and then we can calculate distances we can calculate these functions over a range of distances to address one of these challenges where we're not sure what distances to look over. The K function also has a nice cousin called the inhomogeneous K function, and we can use this to get around the tissue structure issues that we generally face. And so if you were to take any particular pair of cell types, here we've got some gamma cells and some T helper cells, calculate an association statistic. This is the type of information that you might see. And so here there's a box plot where every single dot is a different image, and we've got multiple subjects or patients. And you can see here that the localization in the non-diabetics, these two cells are probably avoiding each other. And then as you get early onset diabetes, these cells tend to mix a little bit more and that structure disappears. And so this is fine to quantify visually, but obviously we might want to test this to construct some sort of p-value. Reading the literature, we've seen people simply ignore the subject information altogether and treat all the images as independent and do a t-test. This is obviously a big no-no because you're violating your independence assumptions. We've also seen people just average these similar localization scores within each person and then compare these, but you're throwing away information. And so instead we've started analyzing these things with a mixed effects model. This is a standard thing to do when you've got repeated measures. And so we use this to account for the challenge of having multiple images per person. While I said that these localization statistics, the K function and the L function and the derivatives of this, the scaling makes these scores comparable across images. There's still a summary statistic. And so you've effectively got an average score for each image and you've thrown away other information in that image. Specifically, you've thrown away the information about the size of the image or how many cells are there. Which gives you some measure or feeling of the variability or the reliability of your scores. And so here we can see a scatter plot. Each dot is a particular image, and we've got the localization statistic, and we've got the number of a particular cell type in each image. And you can see that the images with low cell type, the scores are highly variable, and that variability decreases the more cells you get in the image. And this makes sense. The more cells you have, the more information you have, the less variable a measure should be. And so this seemed like really valuable information that we could leverage. And so we simply take effectively share information across the summary scores of all of these images, fit some curves through some squared likelihoods and use these as weights to either upweight or downweight an image in our mixed effects model, depending on how reliable we feel they are. Because we're testing things at least between pairs of cell types, these aren't just simple scatter plots like we saw there, they're bivariate, and we fit kind of weight curves in this two-dimensional space. And so we've been having a lot of success with this approach. Like any method that you develop, you can always construct some sort of simulation that demonstrates that it's the type of thing you would like to use. And that's what we can do. We can generate simulations and show that including these weight scores is useful. And so again, I'd like to thank everyone for the opportunity to speak today. And more importantly, I'd really like to advertise that I really love talking to people. And so down here in Sydney, I'd love for people to either, we're kind of locked out at the moment. The borders are shut, and when they open, come down and speak to me, or more importantly, reach out on Twitter, and I'd love to have a chat about things. And so thank you very much.
So I had a question, which was how many cells are enough for your point process analysis to be reliable? That means detect as similar things that are similar and detect as different things that are different.
Well, I guess it really depends. It depends on not just the number of cells, but also the size of the image. And I guess the amount of images you've also got as well. And so if you've got lots and lots and lots of images, then even if you've only got a small number of cells in an image, you can kind of be confident in the aggregate. So there's no magic number, but
okay. But in thinking of using point processes as a way to distinguish, because let's say the beauty of it is, of course, you are reducing the cells to points and you're using the point patterns basically to guide you. But it was unclear to me whether you've tested, how many cells are the minimum, bare minimum that you could use to tell that a tissue is the same tissue type. you have any idea for that? Are we talking about hundreds of thousands or tens of thousands?
So this is one of those statistical issues, right? Is that you're talking about testing a null hypothesis,
Sure.
as opposed to nearly we're looking for the alternate. And so it's, if you don't see a difference, who's to say you're just not powered to see the difference, or there's no difference there? That's a really interesting point. And I think maybe even just going and doing some simulations might help address that.
Yes.